She's just brilliant in providing insights and strategies for how to be better organized with your data, how to maintain it, to keep integrity in your data. And what I love most about her is for all of us that think we already do that, before the day is over, you will realize that you can do it better.
How many people do I hear have, that actually witnessed you say that? This is great. It's recorded. All right. Hello, everyone. My name is Melissa Eitzen. I have the honor of talking to you right after lunch, right? So this is what I'm going to talk to you about today. I'm going to spend the first, not quite half, talking about some of the principles, guidelines, policies, and definitions. You will have a copy of these slides. My goal is twofold. Number one, I have a mentor that told me years ago that common sense is not as common as you might think. Right? So if you remember Dr. Baggerly's, our intuition with big data sucks. And with me, common sense is not as common as you might think. So my job here is just to open up your brain, open up your mind to a little bit more awareness to think about things. I'm not an expert in all of the different scientific disciplines, but what I teach typically will be agnostic, and hopefully you can apply it. So I will give you some tools just so that you'll have them as a reference. You will have these slides available to you after today. And then I want to spend the second half really talking about the data life cycle, giving you some tips that hopefully will be helpful to you. Then we'll do a breakout for a case study. It's a very simple case study. There's no right or wrong. And you guys will report back and we'll talk about it. So we talked, I think, earlier about the regulatory pathway. This is my home. If you're not familiar, this is an oversimplistic diagram. I find that more people than not don't appreciate where discovery science is related to products that are approved by the FDA to better human health. So on the left side of your screen is what we call discovery research. From that, and I don't mean to oversimplify it, that's a lot of work, but from that if you're translating your science, you would move to non-clinical efficacy to prove that your product, your vaccine, your therapeutic works. And then you enter what we call the regulatory pipeline, where you must prove very robustly under FDA regulations that your product is safe. Because we don't care if it's efficacious if it's not safe. That's important. Those where the regulations kick in. From there, then the FDA makes a decision about whether or not they give the sponsor permission to go into clinical trials. So it's phase 1 or healthy population, those are your safety studies. Phase 2 are the smaller groups. Phase 3 get larger and larger. Then again, there's another regulatory decision that the FDA makes to approve. And then finally, it moves into post-marketing, which in some cases, there's a phase 4 clinical trials. There are subdivisions of all of this. So for anybody that does clinical research, I do not mean to offend you. This is just a broad stroke to get you folks to understand where the pipeline is. In your basic science or discovery science, for those of us who have been around long enough, we call this self-correcting. So we publish our research, and our reputation went into that publication. And if something was wrong, we would be called out on it. And that was really sort of this self-correcting peer-to-peer review that took care of the quality of the science. The last time that I did the numbers, which now has been, I think, 5 or 6 years ago, there were over 8,000 publications a day. And I know there's more than that now. So how do we keep up? So it's really impossible to do that checks and balances just with publications alone. And there's really no regulation that guides discovery research because it's too broad stroked. We're still holistically thinking about things as it should be. There are some quality guidelines out there, but there's nothing that says, other than peripheral regulations on whatever we're doing. So there's select agent or there's animal welfare, but there's nothing out there that really talks about the quality and integrity of our data that's a regulation. I like this article, although I don't speak to it, but it really shows and illustrates that there are 4 pillars or 4 stakeholders in data quality and integrity or robust science. That's the funding agencies. So there's some responsibility there in terms of if they give you money, then they want to have some assurance in the quality and integrity of the data that you're producing. The publishers, there's really some onus on them, and the researchers, which you are in the room, and then the institutions. I'm not going to talk about the institutions today. That'd be a whole nother lecture. This particular article, you can see the researchers looking at the institutions like, help me, help me, help me. And I think in some cases they are trying to do that, but that's another lecture for another day. But I just want you to understand, for me, I want to just touch on those other three. So in 2014, just a little bit of history, there was the NIH, which is the largest funder of public research in the United States. They got together with two publishers, both Nature and Science, which are the top journal or publishers of NIH-funded research. And they said, what can we do as the funding agencies and of the publishing agencies, what can we do to help encourage rigor and reproducibility and quality and integrity of our data? And they got together. There were over 30 science journals represented there. And what they wanted to do was they wanted to try to do something to enhance rigor and further support research that's reproducible, robust, and transparent. Their outcome was a set of principles to facilitate these goals, which would be, hopefully, a number of journals would agree to endorse. This is the principles and the guidelines that came out of that particular workshop. And this should start to look familiar to you, because if you're putting in for NIH grants and you look and you see what the requirements are, these things you see now. You have to address them. So rigorous statistical analysis. We heard Dr. Baggerly talk about, errors that went terribly wrong over something that was very simple. Transparency and reporting all of your data, even the outliers. Because sometimes you don't know why something is an outlier until the science evolves. Data and material sharing, best practices for images, manipulation of images, biological materials, of course, our animals, and having journals endorse these and having adapted guidelines based off of these principles. So my job is to really narrow in and focus on data and material sharing. And of that, they agreed that the data set should be made available where it's ethically appropriate, both during manuscript review but also upon publication. So at any point in time, that was the goal, was that if you are taking public money, you have an obligation to keep that data and to share that data. They also recommended that these data sets be public. You're seeing more and more of that with the journals. They're saying, if you're going to publish, we want you to either tell us where the data sets are, or they have repositories that you can use to store your data. Encourage presentation of all your data. And I know we'd all love to publish our negative data. I think everybody I've ever talked to says, gosh, well, I wish we could publish our negative data, because we learn just as much from our, I don't want to say failures, but we learn just as much from experiments that don't yield results that we thought they would than we do from those that confirm. So we want to make sure that the data are also readable. There's code available that you can even find the code. Sometimes these are homegrown, and that can maybe even done by a postdoc, and then the postdoc leaves, and then the PI that's there 3 years later gets asked for the information, and the PI doesn't know. Research is transient. Labs are transient. People come in and people leave. And not always is there some kind of a continuum there that we can actually grab hold of things, particularly after a few years pass by. And require materials sharing after publication, including software, and then just a statement that says, if you weren't able to publish all of your data, where is it? How do you have access to it? So that was 2014. From this, you started to see the NIH set up policies, which is probably partly why you guys are here. But the purpose is really that the data sharing enables researchers to rigorously test the validity of research findings. It strengthens the analysis through combined data sets, allows you to reuse hard-to-generate data, and explore new frontiers of discovery. Sounds exciting, doesn't it? Like it should scroll through like there's a movie or something that's coming. In addition, NIH emphasizes the importance of good data management. And this is why I'm going to talk to you about data management, because we talk a lot about the data, but we don't think about being stewards of our data all the way through the process. We just hurry and collect it. So, anybody heard of the FAIR principles? Yeah, this is on the NIH website. It's really good. I would, if you're writing your data sharing, management and sharing plan, you might want to Explain how your data are going to be organized such that they're findable. It's probably the hardest part. Accessible, interoperable, which means if you have more than one operating system that they're communicating to one another, and reusable. Now, this is for you guys. I encourage you, if you haven't done it, take a lab meeting and watch this together. It is painfully funny. Anybody watch this? It's painfully funny. So there's an oncologist that goes, reads an article, goes to the scientist and says, oh my gosh, I loved your article, I read it, I really want, XYZ data. And the scientist said, everything you need to know is in the publication. And the oncologist keeps drilling down and asking harder and harder questions that are very logical, but the scientist can't find it. And the more that you go down this rabbit hole, the more that you realize there's no way that they could put their hands on the data that the oncologist was really interested in. And it's comical, but it's painfully comical. So I would encourage you to take a lab meeting, watch it, and then discuss it. So with resource sharing, NIH continues to considers the sharing of unique research resources developed as an important means to enhance the value and further the advancement of research, and they want those results to be readily available. It's public funded research. And I speak a lot to NIH, but it's not just NIH. I have a colleague that's a veterinarian up at the University of Minnesota, and she actually got an award from the Smithsonian. And the Smithsonian had all kinds of quality requirements. If anybody here has gone in on big center grants, especially if you're working with like the small business or you're working with partnering with business and industry, there are requirements now from those funding agencies to have even more robust quality systems around your data, besides just saying where you're going to store the data. So I'm starting to see this as a trend in the industry, especially big center grants. It's also important to note that resource sharing goes beyond data. It includes things like samples, if you're generating samples, reagents, model organisms such as transgenic mouse strain. But for me, I'm going to focus on the scientific data. The policy definition of scientific data, I know this sounds boring, but it's actually pretty important. The recorded factual material commonly accepted in the scientific community as of sufficient quality to validate and replicate research findings, regardless of whether the data were used to support scholarly publications. So even if you don't publish, the NIH expects you to share your data or have a data plan that says how you could share it should someone ask. Scientific data, according to the NIH policy on data sharing, do not include things like your laboratory notebooks and preliminary analysis and such things that are listed here. I think it's important as researchers to realize that you do have some ownness of the inner workings of your laboratory that you're not required to share from a data sharing perspective. I will caveat that by saying, if you're working on the regulatory pathway, the closer that you get to having something that needs to be regulated, the more you're going to have sponsors ask you to retain all of that information, because that's part of what's needed to reproduce. That's part of what's needed for your study to be transparent. So if you have a good lab manager or a good lab management system, you might have, I would hope you would have, the ability to organize a lot of this, because it is going to be important for you to go back and reproduce what you did or have someone else be able to reproduce it. But as far as data sharing from the NIH, these are the things that you don't have to include in your data sharing plan. Just a note. I don't know if anybody works off of business contracts. Anybody work off not just grants, but contracts? Because contracts are a whole nother thing. We get into contracts at UTMB and our office, particularly from pharmaceutical companies, and they want us to run a product through an animal model, and part of that data might actually be submitted to the FDA as part of a submission package. And because it's a contract, there will be some additional things that they want us to keep. So just kind of a courtesy reminder to you, when you move from grants to a contract, read the contract. I have another really good colleague, very well known in the field, who took money from a contract. It was a pharmaceutical company. And his opinion was the contract was all legal jumble, and the lawyers were going to take care of that. And it was through, in that contract, he'd agreed to being audited by the sponsor. And the sponsor shows up to audit the data to say, okay, where's the proof to show that, you know, this data actually happened, blah, blah, blah. He's like, well, you can't audit me. And they're like, it's in the contract. Where in the contract? I thought that was the law, you know, the legal contract. So be sure if you enter into any kind of contracted studies that you pay attention to the language. That's just my side note. Metadata, I think Dr. Bagley talked about this too, but metadata are any data that you need that explains your results, your data. So there's a lot of metadata in terms of when something was collected, who was it collected by, that's important. But there's also metadata that support collection or analysis of samples where there's a lot of metadata that go into that final result, and that's also important because you can't reproduce it without all of that information. So you need to be thinking about that too. So data management then is the process of validating and organizing and protecting the data all throughout its life cycle. Data sharing, of course, is how do you make that available to your peers? And data management and sharing plan, which is now required on your NIH applications, is just a plan that describes how you intend to do that. So where do we begin? This is the second half of my presentation. I like to look at data through a data life cycle. I'm a visual learner, so I like to look at the data life cycle. And the data life cycle starts when you collect your data, but it continues to, how are you going to organize and store it until you analyze it? Once you analyze it, where are you going to retain it? How are you going to share it? And how could somebody potentially reuse it? We don't talk about disposition here because that's really the institution's responsibility or the state of Texas, for those of us who are here in Texas. But I think what I see is we're so focused on collecting the data and analyzing the data, we don't think about how we're organizing and storing it, and that can impact our quality and integrity of the data. Certainly, the FAIR standards, it's not going to be findable if we don't know how to organize it. So I pulled this from the University of Pittsburgh. This is from their library, and I really like it. Data are a scholarly product. That's your product. They're fragile, they're easily lost. There are growing research data requirements, and good management helps prevent those errors and losses and increases the quality of your analysis. Well-managed and accessible data, again, those FAIR standards, allows others to validate and replicate findings, and your research data management facilitates that whole process. So I just want to kind of walk through this life cycle and give you some ideas. The first thing you want to do in a data sharing plan is you want to do an inventory. So prospectively, what kind of data are you going to expect to produce? What kind of format? What's the quantity? Because you can't really wrap your brains around how you're going to organize it if you don't understand what you're producing, particularly if there are multiple PIs or multiple scientists that are working with you. So do an inventory, do a prospective inventory about it. Think if there are any other standards with which you need to comply. So if it's clinical data, you've got the HIPAA standards or anything else that you might have. If you're partnering with business, are there any proprietary concerns? That gets really important because NIH, you're accepting NIH data, once you get funded, right, then you open yourselves up to the Freedom of Information Act. And if you have anything that's proprietary, then you have an argument for your legal team to defend and say, this is proprietary, we can't release X, Y, Z. So you want to get ahead of that too. And more often than not, I don't see people identify who the owner is. So who do you go to if you have a question? Especially if it's a multi-center or multi-scientist are involved. Who do you go to? Dr. So-and-so is going to be the lead person, contact person for X. Versus Dr. such and such is going to be the lead for all the PCR data, for example. So let's talk about source or raw data. Raw data, source data is the first time it's captured, whether it's written down, whether it's notating in a lab notebook, whether it's an electronic notebook, a paper notebook, a field notebook. Maybe you're manually counting plaques up there on the top left and you're writing on it. That first capture is your original raw data. Your original raw data, this is an industry thing, and I really like it. If you think about data quality, it should be attributable, legible, contemporaneous, original, and accurate. Attributable, again, who do I go ask if I have a question? If I don't know who recorded that data, I don't know who to go to. Legible is kind of self-explanatory. You need to be able to read it, but that also means you have the code to be able to interpret it. Contemporaneous. How many people remember what they had for dinner last Wednesday night? Yeah. So when I was in a research lab, we used to do all of our studies Monday through Friday at noon, and then Friday afternoons we're writing in our notebooks. So Friday morning, anything I did on Thursday was pretty meticulously written, and anything that happened on Monday, Tuesday, and Wednesday was kind of iffy on what I wrote down. Not a good habit. So the sooner that we write things down, the more accurately we capture what happened. It's a very simple, commonsensical thing. And then original and accurate. We'll talk about why I mean original in a minute. And then as an entirety, your data integrity is, are your data sets complete? Is how you're recording and organizing it, is it consistent? Hence your data management plan. Is it enduring? Can I find it? Is it readily available? Data are the foundation on which scientific, engineering, medical knowledge are built or is built, and data integrity is the degree to which data are complete, consistent, accurate, trustworthy. I just gave a lecture in an ethics course 2 days ago, and they were given 3 or 4 different articles to read, and one of them was the Ellis article on irreproducibility. And trust just kept coming up and up and up, over and over and over again, trusting your colleagues. Reliable, and these characteristics of the data are maintained throughout the data life cycle. Look familiar? I know, this is what we do. I do it at home. Somebody calls, I grab the first piece of paper, I write it down. Scenario 1 is the worst case scenario, it gets put in the trash. It's happened, right? It's gone. Maybe I can go take the pH again. Scenario 2 is I transcribe it, and it goes in the trash. Scenario 3, which is a lower risk, is, okay, I'm going to either take a picture of it and put it in an electronic file, or I'm going to tape it to my notebook. Scenario 4 is eliminate that bad practice. Please try not to do that. Same thing with when I'm working at a hood. I used to do all my math on the cabinet. Yeah. So here's a transcription error. I have a hematology, you probably can't see it in the back, but on the left, it's a hematology printout. And in this example, it has a hemoglobin of 13.6, which for those of you who are clinicians understand that that's a normal for a human and normal hemoglobin. But when it got transposed in this hypothetical situation, the 1 got dropped. Sticky key, something happens, and now my normal goes to an abnormal. If you have a hunch and you look at it and you go, why is that one so much lower? I wonder if that's accurate. What's the first thing you're going to do? You're going to want to go back to the original data and double check it. If you don't keep that original, you have no way of going back and determining whether or not that was a systematic error that was just due to somebody transcribing it incorrectly. It had nothing to do with the science or the assay itself. All right, how many people think this is May 21st? Show of hands. May 24th. 27th? 29th. About half and half. This is my son's handwriting. Should have known he was going to be a dentist because he can't write, still can't write, but he can read it. Yes, I did get his permission to use this. We were actually freezing down some chicken broth in the kitchen. And because I'm a long-term laboratorian, got my undergraduate degree in It was called medical technology. It's called medical laboratory science now. We dated everything. So if you open up my freezer's dated, because I learned this when I was, 20. If I open this up in my lab and I saw this date, would I care whether it was the 24th or the 27th or the 29th? Probably not. I care that it's 2011, right? Probably going to pitch it. For sure I'm going to pitch it if it's chicken broth. But if I was in the process of mixing this and it had a stability of 24 hours, it now makes a difference whether that's the 24th, the 27th, the 29th, or whatever number it is. So legibility can be a big deal. How many people think this is May 3rd, this date? How many people think it's March 5th? Yeah, it depends, doesn't it? So think about, we're multicultural, which is very cool, but people write down dates differently. Day, month, year, month, day, year. So you might want to think in your lab to talk about, maybe it's MAY for May or MAR for March. Then it doesn't matter what order it's in, you're always going to be able to know it. Try putting in the numbers in an Excel spreadsheet, and it automatically defaults to something, right? And it goes to the English version. Yeah, I feel your pain. It goes to the English version. So just simple things like that, common sense, not as common as you might think. All right, so 30-second exercise just for some fun. Grab a scrap piece of paper like you're in the lab, and I'm going to reveal this little picture of something, and I want you to write down everything that you can to describe it. And you're going to have 30 seconds, so really fast. Ready? Ready, set, go. 30 seconds. $25 seconds. $15 seconds. 5, 4, 3, 2, 1, stop. All right, somebody just shout out. What'd you write? What was that? Brown mouse. Anybody else? Cheeto. A Cheeto. All right. What else? It looks like a Cheeto. Looks like a hot Cheeto. Sitting in someone's hand. 12 whiskers. Anybody else? Anybody else think they're eating a Cheeto or something else? An orange peel. Suzanne. Halloween candy pumpkins. Anybody write like bright-eyed, alert? Yeah, okay, those are my animal people, right? Excited about a Cheeto. Mouse smiling. All right, so, look, see, time's up. So the reason why I say that, I want you to go back and I want you to look, and did anybody make it attributable? Did anybody date and initial it? Probably not. It's original data, right? The reason why I do this exercise is because usually what happens is we get into this debate about what the little guy is holding. Somebody says strawberry, somebody says carrot, somebody says Cheeto. I've not had a Halloween pumpkin, that's a new one. And so the reason why I say this is I don't talk a lot about training, but training is a big deal with data quality and integrity. Because if we're not trained to know what we're looking for, then we're arbitrarily collecting data. Maybe I care that this mouse is alert. Maybe I care that this mouse likes this particular diet. There could be, maybe this mouse is on a study where there's an eye treatment, and I care that the eyes are wide open and bright and not glossed over or not droopy. So training, this is my slide to just say training is also important. Don't forget about it. Storage. So storage and retention, I just put these all on one slide. These are all things that you want to think about. And there was a comment about earlier about systems and having newer versions of systems. So when you think about managing data on a system, on the regulatory side, we always think about data migration and what could potentially happen to data as it migrates through. Or Apple versus Mac. IBM versus Apple. I have another group that I work with CRISPR technology. One of the things that he does is he has a file that he locks down that has all the original data. And the only person that has access to that file is the PI and one senior postdoc fellow. That's it. Then he makes an exact copy of that, and everybody else does all the analysis off the exact copy. And that way, if anything happens or is inadvertently lost, or if the Mac language, because I know there's a computer program that one of the scientists use on that, if there's any kind of problem, they can always go back to the original data. So think about things like protecting, if you're writing in a lab notebook, do you put it up in a cabinet at night or in a file cabinet at night? Because I don't know, I work down at UTMB in Galveston and we get hurricanes. So I don't even leave a lab notebook out on a counter anymore. I put it in some kind of a cabinet just because. All right, so managing electronic data, think about audit trails, whether or not you can turn these on, security and encryption, any kind of backups, impact of significant digits. Several years ago, I was in a board meeting and someone had to borrow a computer and download Skype because we had somebody remotely that was joining through Skype. When they downloaded Skype on that computer, it disabled the copy and paste function of the Excel. And we were all like, what just happened? So sometimes you don't know what the risks are to your data. So again, taking steps to protect it. A couple of examples here. One is a documents library, which is just setting up your experiments the way you organize them the same way over and over and over and over and over again. This is really helpful for graduate students. It's really helpful for somebody else coming in. It says all of my studies are going to be organized in this particular way. All my study plans are going to be in this file. Any compliance documents or approvals like IRB or IACUC are going to be somewhere. However you decide to set it up, you just set it that way and you just duplicate it so that every time you know where you're going to look and how it's going to be organized. The one on the top right, I just pulled that from Google. I thought that was a really good example of a file naming. So you can actually get inventive on how you're going to label your files, especially if you keep adding to an Excel file. You want to make sure that you can always go back to a certain point and double check, because as we saw with Dr. Baggerly, copy and paste errors happen. Be careful of save as, autosave. This Office 365, if it doesn't autosave, is a great thing, but it automatically rewrites over your data. So as soon as you go, oh no, I just put that in the wrong column, it's already written over your data. And you got to call your IT to find out how do you go back and get that other version. So sometimes what's a convenience you need to think about. I usually get asked about electronic notebooks. So I take a slide and talk about it. The pros are, I'm really jealous. I wish I had them. I've aged out of the lab now, so I won't ever work with it. But it really helps with organization and collaboration. The other thing I really like about them is that you can program them so that it won't even let you go past if you haven't collected a particular data point, which is also something about data quality and integrity. If you've got 4 or 5 studies going on, you know, and maybe you've got 15 critical time points on one study and 10 on another one, and they're overlapping, Murphy's Law says you're going to just have human error and forget one. So lab notebooks can be programmed so that they'll make sure they prompt you to say, hey, did you collect this today? Hey, did you collect this today? Or what were your reagent lot numbers? Where did you get it? Did you get it from Sigma? Did you get it from someplace else? So that all of your information is there. You can do timestamps. You can put an audit trail on them so you know they're attributable. You can make them searchable, all those kinds of things. And you can usually export the data. Beautiful things. Now the not so pretty stuff. If you work for business and industry, then it's somebody else's problem. Woohoo! If you work for academia, you always have to think, what happens if the funding source for this particular program goes away? What do I do with my data that's already in there? So I always say, please make sure it's extractable, that you can actually pull it out of your system in some kind of an archivable format, a PDF, an HTML file that's archivable, something where you can pull all that data in the event, especially if you're a graduate student and you haven't defended your dissertation and all of a sudden somebody decides to pull that from you, you're going to lose some sleep. So just remember that somebody really has to be responsible for making sure that it continues, there's funding for it, and there's somebody on the back end that understands the software. So anytime there's a software patch push, there's also some kind of testing to show that it didn't impact the data that's already in there. All right, so moving on, analyzing. I don't spend a lot of time on this because usually there's a lot of different disciplines in here. So I'll just say, if you're doing any kind of biological assay work, let me just share it with you. Please define your inclusion and exclusion criteria. Please. Because this eliminates bias. I see it all the time. You run a sample until you get the result you want. If it's not the result you expect, something has to be wrong, run it again. Run it again. I also see people will say we run a sample in triplicate. And I could ask people you give me different definitions of triplicate. Is it 3 samples? Is it 3 aliquots of a sample? Is it mixed? Is it not mixed? You know, how is your triplicate? And then what's your acceptance rejection criteria if one of those is not within the acceptable limits of your assay? So what's the criteria for rerunning? And if you have an assay that's robust, then you should know what those criteria are. And that helps eliminate that bias. So we talk about outliers and reason for them. You have rules before you ever start doing it. And have written procedures, written protocols for anything that you routinely do, including keeping your kit, you know, like if you're doing a DNA extraction and you're using a QIAGEN kit and it comes with little kit instructions, keep them. Because sometimes they change the kits. Sometimes the reagents in the kits change. Usually it's right in the middle of a study without warning. Been there, done that. All right, image manipulation is a huge one. Keep your native images. Always keep your native ones. Of course you're going to manipulate them. We all do. Make sure that you're not inadvertently deleting something that's important in this cartoon. You can't see the teardrop cells and part of the red blood cells anymore that look like they're low on hemoglobin because they just, the contrast and the saturation were increased, causing the background cells to disappear. So you might make a note on what you did. And then this one is important because this is the case study that we're going to start in just about a minute or two, and we'll have about 6 or 7 minutes to do it and about 2 or 3 minutes to report out. I'm going to give you a really, really simple scenario, and I want you guys as groups to think about what would be the conditions for sharing or not sharing. And that's what you have to get ahead of in your data sharing plan. You have to say, how would I do it? Maybe there's multiple methods. Maybe there's some conditions that I need to put on the data before I'm going to share it. All of this goes up front to the NIH when you're getting funded. And please understand that all of the data has to be agreed to be shared no later than the time of publication or the end of the award support period, whichever comes first. So this goes back to even if you don't publish, NIH intends for you to be able to share it should you be asked. Anybody use import? NIH import? Okay, so this is a tool that's on the NIH website. I know some folks who use it, and this is the table of contents, and no, I don't expect you to read it. It's not an eyesight, but I just wanted you to see some of the examples of what the NIH already has set up as some of their database on what's important. So things like experimental samples, control samples, PCR results, reagent sets, reagent sequencing, standard curves, the treatments. Things like that are all considered raw data sets. All right, a slide here is a reference. People usually ask, can I charge? Can I write a line item in for the expense? And I'll say, please go look at the policy to see what's allowable and what isn't. So my closing thought is, we go back to this article that Begley and Ellis wrote about the 53 landmark studies, but I wanted to say the 6 that were confirmed have these 4 elements in common. They included controls, they validated their reagents, they controlled for the investigator bias, and they described their complete data set. That's the plus side of that. So, my tips to you, get organized. It's the hardest thing you'll ever do. Get organized. Be a data stewardship throughout your data lifecycle. Implement the Alcoa principles for data collection. Verify the requirements in any type of contract or RFP that's up. And remember that for NIH, the data management and sharing plan becomes a term and condition of the award. It's pretty serious now. All right, so here's your case study. I really only have about 5 minutes, but it's a short study. Identify options or conditions for sharing data from a study with 500 human subjects being screened for sexually transmitted diseases. So obviously there's going to be some conditions for sharing on this one. So maybe just give three conditions if you guys could, and then I'll call you back in about 5 minutes and we'll just shout them out. Somebody shout out something that they wrote down. De-identify, absolutely. HIPAA requirements, right? That would be a condition. What else? Consent from the patient. Yeah, that would be in the IRB that would have to be in there, right? Yeah. What else?
Data handling.
Data handling. What about data handling?
Secure data handling, transfer and storage.
Security. So there would be some condition that someone would have to show the security before you would hand it over? Is that what you're saying?
Set up some kind of plan for transferring the data.
Some type of a plan to transfer it over that assures the security of the data. That's good. Someone else? Yes, in the back? Conditions for what they're going to use the data for. There we go. Yeah. Anything else? I'm just going to put this up there. I didn't trip. This is an NIH. This is from an NIH website, and this was just the example that they showed as a positive example of what you could submit for that. So a commitment that using the data only for research purposes and not to identify any individual participant. A commitment to securing the data using an appropriate computer technology, and a commitment to destroy or return the data after analysis are completed. So what I mean by there's no right or wrong answer, but I don't want you to think that you can't defend or get ahead of. You can't just say, oh yeah, you can have my data. There may be some times when you need to put some conditions around it, right? Especially a small study. Yes? Yeah, and I think that's actually another really good condition, which is that you want, maybe that's a condition for sharing because the subjects are so small, maybe there needs to be some cooperation on the publication of it. And maybe what you're saying is you as the primary author are going to retain the original data, but that they need to destroy XYZ because there's some sensitivity involved. I think that's what the point of this was. It wasn't, I always hate to say you destroy data. But if they're working off a copy, it's to protect the subject so there's not copies and copies of it elsewhere that you don't have control over. I think that's that point. So Stacy, if you want to come up and get your slides going, I'll entertain if anybody has any questions while we're doing that. Any questions or any comments for me?
I did want to ask you about
the
slide that you had up about color enhancement of images. Is it okay as long as you put down for the purposes of, focus on, the appropriate cells, the images were, okay.
Absolutely. A descriptor is what you need, right, in the native file. So especially because I have seen where you submit images to a publication and they're going to ask you, was it manipulated? Or sometimes they might even ask you for the native file to make sure that you're not being coercive in terms of what you're presenting, like that you're biased in terms of, oh yeah, look how great this stain was. I've also seen on the Office of Research Integrity, they'll show where people are using controls and then they readjust the image and then falsify it and say it's the sample. Yeah, I mean, there's a lot of falsification around images going on.
See, a lot of times I take images with my cell phone, and then it may be too dark, and so then I lighten it up a little bit, and I'm always kind of, you know, hesitant. Do I
Keep
need
the original.
Yeah.
Keep the original,
Okay.
for sure. Yeah, comment.
On that microscopic topic, what about manipulating the natural intensity or the setup of the microscope, or image?
Yeah, I mean, so equipment I don't talk about, but equipment maintenance is a whole nother big thing, right? Including, Keith was talking about, you know, a 96 well plate and the fact that there wasn't even temperature and it cooked the cells. So even thinking about, you know, quality control and what's going on with your piece of equipment, for sure you want to record the magnification, right?
I guess the native file will have that type of info.
It could. You might double check.